Hi, everyone. Today, as our guest, we have Professor Vatsala Thirumalai, who is a professor and dean at the National Center for Biological Sciences at the Tata Institute in Bangalore, India. She's in fact presenting this talk to us from India right now. Her group studies how the brain generates locomotion using the larval zebrafish. So she received her PhD with Eve Marder at Brandeis University, then did a postdoc with Holly Klein at Cold Spring Harbor, then returned to India to set up her own lab. She uses electrophysiology and imaging techniques and genome editing to look at questions regarding control of behavior. She serves on the editorial boards of eLife, Journal of Physiology, and a number of other journals. She is the recipient of a number of awards from the government of India, and she's also been awarded the Senior Fellowship of the Wellcome Trust India Alliance. Vatsala, thank you so much for taking the time to present your work.
Well, thank you very much for the invitation, Reza, and it's always a pleasure to present this work in front of a cerebellum audience. So it's my great pleasure to be here tonight. So this work that I'm going to talk about is actually the PhD thesis work of Shivangi Verma, who's wrapping up her thesis, and we are actually writing this up. And so I appreciate any and all feedback on this work, which will make us, fine-tune the manuscript much better. So thank you very much for the timely invitation. So if I may go ahead then. So as Reza mentioned, I work at the National Center for Biological Sciences in Bangalore, India, and that's our beautiful campus. I urge you all to find some opportunity to please do come and visit us. It's a fantastic campus with a range of biology, from molecular biology, neuroscience, to ecology and evolution. So if ever you are in India, please do drop me a line. So let's dive into the work. So we all know that synapse elimination is as much a part of circuit maturation as synaptogenesis. And the brain forms more number of synapses than it needs in the mature condition. And in humans, for example, there are two major phases of synapse elimination that are known. One is in the toddler years, early synaptic pruning, and another one that occurs in the adolescence. And it's known that if synapse elimination is impaired in either one of these periods, it can lead to major neurological conditions such as autism spectrum disorders or schizophrenia. And we know a lot of different places where synapse elimination happens in the nervous system, starting from the periphery, where early on in life, individual muscle fibers are innervated by multiple motor neurons, to later on, where every muscle fiber receives inputs from only a single motor neuron, and the rest of the inputs, supernumerary inputs, are pruned away. It's also known in the visual system, where in the LGN, eye dominant territories are established by synapse elimination. But for this audience especially, and because it's a topic that my lab works on, we will focus on synapse elimination in the cerebellum, where it is known that in neonates, the Purkinje neurons of the cerebellum are innervated by multiple climbing fibers, which are axons originating from the inferior olive. And later on, in rodents, for example, this happens in the first 2 weeks of life. These multiple inputs from the climbing fibers are pruned such that only one climbing fiber remains. And in the adult, every Purkinje neurons receives only a single climbing fiber input. So we know a lot about the mechanisms of synaptic pruning, the developmental mechanisms, both molecular and cellular. We know the way in which the climbing fibers slowly migrate towards the proximal dendrite, as shown here. And we also know the molecular mechanisms by which the other climbing fibers are pruned away, and molecules have been implicated, such as calcium channels, metabotropic glutamate receptors, various different kinases, etc., etc. But what is less clear is why the circuit undergoes this sort of maturation process and what it really does to the circuit output and behavior. We all know that toddlers keep asking, but why, but why? And so with a toddler-like curiosity, we approached this question and asked what it means for the cerebellar circuit when it undergoes this massive synaptic elimination, particularly because these climbing fibers are among the strongest synapses known in the central nervous system. So when you remove such strong synapses, it obviously must have some major impact on circuit function and behavior. So we asked, how does cerebellar synapse elimination affect circuit function and behavior? Now, there's one problem though, which is that in the mammalian system, this cerebellar synapse elimination happens during developmental stages where motor behaviors are very limited. For example, in rodents, as I said, it happens in the first 2 weeks of life, where the pups are initially, their eyes are not open, and even after their eyes open, they're not able to balance their body weight, they don't move around much, and there's not much motor behaviors to speak of. So how can you assay the effect of cerebellar synapse elimination on circuit function and behavior when behaviors are absent? And that's where the larval zebrafish comes into play. Larval zebrafish can be used to study this problem for many reasons. Number one is the shared evolutionary history where cerebellar circuits are conserved from fish all the way to mammals. So in zebrafish, Purkinje neurons are present, climbing fibers are present, inferior olive, all of the different circuit elements are present and the connectivity remains more or less the same. And we, documented many of these properties in our paper published in 2015. And also that because it's a fish, the fertilization is external and many of the early developmental stages are accessible for study, unlike an in utero development where early life stages may not be accessible for experimental manipulation. The other major factor that helped us study this question in zebrafish is the fact that these organisms are independent as early as 5 days post-fertilization. So I'll keep using DPF everywhere. DPF stands for days post-fertilization. And even as early as 5 days after fertilization, these organisms need to find their food and they need to stabilize themselves in the environment around them. So they exhibit a number of different reflexes as well as goal-directed behavior to catch prey. And they're able to respond to applied sensory stimulus. As you can see in this video here, this is a 5-day-old zebrafish larva swimming around and exploring its environment. I'll show you more behaviors that we use in the lab to assay cerebellar function. The other factor is that even though the animal is independent and behaving, during this time the cerebellum is still developing, it's still being put together, as shown by studies both from our lab as well as other labs. Tamily Weisman's lab showed that Purkinje neurons are born even up until about 8 days post-fertilization, and then it sort of stabilizes at a number of around 300. We showed in our lab that Purkinje neurons are elaborating their dendrites, and their dendrites grow in complexity and branch numbers through these days, 5 days through 8 days. So the cerebellum is very much immature and developing. The animal is behaving, and the cerebellum is activated during these motor behaviors. So both work from our lab as well as work from Misha Ahrens's lab from Genelia. So Misha Ahrens first showed using imaging, and we show using electrophysiology, that if you were to monitor both motor behaviors as well as the activity of Purkinje neurons, you see that they are very much activated during motor bouts. So in our experiment, what we did is, this is a paralyzed larva, and we are recording single cell patch clamp recordings from Purkinje neurons in the cerebellum while simultaneously monitoring fictive motor patterns by applying extracellular electrodes to the skeletal muscles. And the top trace is the voltage trace of the Purkinje neuron, and the bottom trace is the motor bout as recorded by this extracellular recording electrode. And you can see that every time there is a motor bout, which corresponds to a swim bout, if the larvae were not paralyzed, and therefore these are fictive swim bouts, every time there is a fictive swim bout, there is a burst of activity in the Purkinje neurons. And in fish, we see both climbing fiber inputs as well as simple spikes. So the short stunted activity that you see here are the simple spikes, and the tall ones are the climbing fiber inputs. These are unlike the mammalian complex spikes because there are no spikelets riding on top of them, but they are very much akin in function in the sense that they do let calcium in during the climbing fiber. More on that a little bit later. But these are homologous activity patterns between fish and mammals. So to summarize this part of the talk, I've shown you that the larval zebrafish cerebellum is functional even when immature and participates in behaviors. So the first thing that we had to clear was whether there was CFP and synapse pruning in larval zebrafish, similar to what happens in mammals. And if it happens, then what is the period during which synapse pruning happens? So if you look in a 5-day-old larva, you see climbing fibers innervating Purkinje neurons. You can see that even as early as 4 days post-fertilization. Purkinje neurons begin to be specified by about 2.5, 3 days post-fertilization. So this is soon after being born. They are receiving these inputs, and our earlier recordings show that these inputs are quite functional. And if you were to record from 4-day-old larvae, you do see climbing fiber EPSPs, excitatory postsynaptic potentials, even as early as 4 days post-fertilization. So to see whether there is synaptic pruning, first we recorded spontaneous activity from these neurons, and we saw that we saw two things. One is that the frequency of the climbing fiber input. So here we are doing voltage clamp recordings from Purkinje neurons. So the downward blips are synaptic currents. You'll see very small synaptic currents and very, very large synaptic currents. And these large synaptic currents can go up to about 600 to 800 picoamps, which is very large considering the size of these cells. And we see that the frequency is higher in the early stage larvae, whereas in the late stage larvae, the frequency is lower. The other thing that we observed is that the variability in the amplitude of these inputs decreases as the animal matures from 4 days to about 12 days. So these two observations can be explained if we consider that perhaps climbing fiber inputs are pruned away such that one very large synaptic input is maintained while all other weaker inputs are pruned away. And so consistent with that, if we normalize the CF amplitude to its maximum, so 0 being minimum and 1 being maximum, in the early stage larva, which is the 4 to 5-day-old larva, there's a wide spread of amplitudes, both weak ones as well as very strong ones. Whereas in the late stage larva, preferentially, the weaker inputs seem to be lost or absent, whereas the strong inputs are present. So this again is consistent with our view that perhaps during this time from 4 to 5 days old to about 11 to 14 days old, that this CF synapse pruning could be happening. So how do we confirm that? We used a test that is widely used in the field for studying rodent CFP and synapse pruning, published by Masanobu Kano. So what you do is you apply electrical stimulus to the climbing fiber bundle. If the Purkinje neuron were innervated by only one climbing fiber, and you gradually increase the stimulus intensity, at very weak stimulus intensities, you'll get only failures, no fiber being recruited, and therefore no synaptic input. But as you gradually increase the stimulus intensity, you'll get a fiber being recruited and spiking, and that spike in turn leading to an EPSC in the Purkinje neuron. And if you increase the stimulus intensity even further, you lose all the failures and you'll be left only with the axon that's innervating the Purkinje neuron, giving rise to the single amplitude EPSC that you see here. Contrast this with the poly-innervated Purkinje neuron, where multiple CF fibers innervated, each having its own synaptic strength. So initially you do get failures, and then you start recruiting 1, 2, and 3 axonal fibers, and correspondingly you will get multiple steps of inputs as shown here. And then when you reach a maximal stimulus strength, you'll keep recruiting all of them all the time, and therefore you'll get a single step. So what you can do is count the number of steps that you get maximally in each of these conditions, and that will tell you the innervation status. If a Purkinje neuron is mono-innervated, you get a single step, whereas if it is poly-innervated, you get more than one step. And so we counted the number of CF steps at each of these developmental stages, the 4 to 5 day old larva, the intermediate stage larva, and then the late stage larva, which is the 11 to 12, 11 to 14 day old larva. And you do see that in the early stage larva, you see a few Purkinje neurons which are mono-innervated, that is only a single CF step, but equal proportions of 2 and 3 step Purkinje neurons. But in the late stage larva, you get a predominance of the single step Purkinje neurons, which are mono-innervated, and some proportion of the dual innervation. Now, it's worth mentioning here that a recent paper does show that even in the adult, in mammals, some Purkinje neurons could retain 2 CF inputs instead of 1 CF input. So this could be akin to that situation.
Vatsala, may I ask a question?
Yeah.
The fact that the inferior olive neurons are electrically coupled, how does that affect your analysis?
Yeah, so what we did was also to look at the spiking. We recorded directly from inferior olivary neurons. So I have that here. There. Okay, that's the next slide. Okay, so good question. We looked at the frequency of firing of inferior olivary neurons to make sure that, this is not a presynaptic effect. And if they were, there was a difference in electrical coupling that was contributing to this phenotype that we're seeing, then you would see much more frequent firing in the early stages compared to the late stages. At least that was our reasoning. Because being electrically coupled means that the network would fire more coherently during the early stages, but we did not see a difference in the mean spike frequency of the olivary neurons. That is shown here. And we also did not see, so these climbing fibers in fish also exhibit paired pulse depression, just like the mammalian synapses, and the paired pulse depression ratios also did not change, arguing for no change in probability of release of these synapses. Does that answer your question?
Thank you.
Yeah. Okay. So, okay. So there is synaptic pruning happening between these two stages. And the other observation we made was that even in cases where there are two inputs, the disparity in amplitudes between those two inputs was greater in the late stage larva compared to the early stage larva, where the first maximal amplitude and the next one were much more comparable in the early stage larva. In the late stage larva, if there are two steps, the differences in amplitude between those two inputs was much, much greater, suggesting that perhaps this weak input was on its way of being weakened further and being eliminated from the circuit. Okay, having established, so that is summary for this part of the talk, that larval zebrafish Purkinje neurons are multiply innervated by climbing fibers during early life, and that in the second week of life, Purkinje neurons are predominantly innervated by only one climbing fiber, and that the one that is maintained has the highest amplitude, while the others are gradually weakened and removed. This is very much reminiscent of what has been reported in the mammalian literature. And so this gave us confidence that we can push ahead and look at the behavioral and circuit implications of synapse pruning, which is what we set out to do in the first place, right? How does cerebellar synapse elimination affect circuit function and behavior? So there's going to be three aspects that I'm going to be talking about today. One is regarding the sensory tuning of CF inputs, and the second is regarding the motor error tuning. And the third will be to put the two together and talk about how these two changes in the tuning to the sensory and motor aspects leads to a differential a predictive mechanism, and therefore, better performance in the older larvae compared to the younger larvae. So let's get on with it. So I told you that by 5 days, these larvae are independent organisms, and they show a number of different behaviors. The one behavior that we like to use in the lab is called the optomotor behavior. So these larvae in the wild, they live in a shallow stream with very slow currents, and they use optic flow to determine whether they are staying in the same place or if the water current is washing them away. So if they see that the visual scenery recedes from them in a tail to head direction, they move forward, because that implies to them that they are being washed backwards by the water current. Sort of like imagining yourself standing on a treadmill, and the treadmill is moving while you're not moving, and the world around you moves away, recedes, and therefore you have to move forward to maintain the visual scenery around you. So in a free swimming larva, it looks like this. Here we are presenting black and white bars. You can see them as faint gray bars because we've filtered it out to be able to see the larva better. And the larva moves in the direction of the movement of the grating. So the grating is spinning in a clockwise direction, and you can see that the larva is also swimming along with the grating so that it maintains position with respect to the grating bars. We can recreate this in a head restrained larva where the tail is free to move, or we can paralyze the larva and record fictively. Here I'm showing you a tail free larva with head stuck, and you show the black and white gratings, and it will start swimming, moving its tail, and you can look at, if you're imaging, this is simpler. You image the cerebellum and you look at the activity of Purkinje neurons. If you're recording from the Purkinje neuron, then we paralyze it and we use the fictive recordings to assay the motor behavior. But showing black and white gratings moving in a tail to head direction induces forward swimming in these larvae. Now, it's not just the forward direction though. If you can show gratings in the left to right or the right to left direction, and the larva will turn because the sideways movement also induces the optomotor response, and the larva will want to maintain its position by turning in the direction of the grating movement. The group of Ruben Portugues published earlier this year that inferior olive neurons are direction selective early on in development. So in this experiment, they were imaging from the inferior olive neurons, and they showed gratings of different directions, going forward, going backward, going sideways, and in various angles and so on. And they showed that the responses of the inferior olive neurons are directionally tuned. So this is a very important result. And we asked whether synaptic pruning affects how these inferior olive neurons communicate direction information to the Purkinje neurons. So we can imagine a situation where, olivary neurons, which are directionally tuned differently, for example, to directions 1, 2, 3, and 4, all impinge on the same Purkinje neuron, and then synaptic pruning leaves only one of them innervating the Purkinje neuron, in which case the directional tuning of the Purkinje neuron will change before pruning to after pruning. Or the alternate situation is that somehow all olivary neurons tuned to the same direction will impinge on a given Purkinje neuron, and some other molecular mechanism then determines which of those directionally selective neurons remain, right? In that case, the directional tuning of the Purkinje neuron will not change. So we wanted to investigate this and ask if Purkinje neuron directional selectivity changes with synaptic pruning. So we chose our stages where we had earlier established that there was a change in synaptic pruning. So the same 4 to 5 day old larvae versus the late stage, which is the 11 to 12 day old larvae. And again, like in Ruben Portuguese's experiment, we showed different directions of these gratings, the 8 different directions. And now we are recording extracellularly, loose patch recordings. Again, the small blips are the simple spikes, and the large blips marked by these open and closed circles are the CF inputs. And we see that, there in a direction non-selective cell, the CF inputs occur indiscriminate of the direction of the grating being shown, whereas in a direction selective neuron, the CF inputs occur preferentially for certain directions and not for the orthogonal directions. And as we expected, the directional tuning was much better in the late stage larvae compared to the early stage larva. So here we've calculated a direction selectivity index based on the responses, the CF responses for a given direction compared to other directions. And we see that, so this is a polar plot. The amplitude is the amplitude of the direction selectivity index, and the direction of the vector is the actual direction of the gratings being shown. So you can see that the amplitudes of the DSIs are much greater in the early and the intermediate stage larvae compared to the early stage larva. So the tuning to different directions of gratings being shown is altered as a result of synaptic pruning. And because these are CF inputs that we are looking at, we can infer that this is a direct fallout of the elimination of supernumerary CF inputs to Purkinje neurons. So with this, we conclude that CF inputs to PNs are more directionally tuned after pruning. Yeah.
But Salam, may I ask a question?
Yes.
The development would also alter the response properties of neurons in various parts of the brain to that motion. So you might imagine that the input to the inferior olive regarding that motion is also becoming more potentially selective. So there's development there. And I'm wondering, does the response that you see in the Purkinje cells, can you tell whether individual climbing fibers are changing their selectivity, or is it that together they're producing this mixed selectivity early on versus later?
Very good question. So this result has to be taken in the context of the earlier result, where we see the steps, right, which argue for a single strong input as opposed. So these cells, right? I mean, about 70% of Purkinje neurons that we record from have only a single step, arguing for a single climbing fiber, right? I do not deny that there might also be a developmental change in how the olivary neurons respond. But what is indisputable is that, we are looking at Purkinje neurons, which are predominantly mono-innervated, and therefore that is bound to reflect in the change in the direction selectivity index.
Thank you.
Okay, so the next thing that we looked at is, of course, the motor error tuning of CF inputs, which is also known to occur. So in this case, what we did was, again, to resort to the optomotor response, but we gave the gratings in only one direction, which is the tail to head direction or the forward direction. And we gave it an open loop, which means that the fish response does not alter the sensory stimulation being given. Now, in open loop, the fish expects that its own swimming will have an effect on the environment, on the visual stimulus that it sees. And when that does not occur, it leads to a sensory-motor mismatch, leading to an error signal. And so what we did was, in this case, we did voltage clamp analysis, because that allows us to assay the strength of the synapses in addition to the timing of those responses with respect to swims. So we are recording fictive swims in this case. That is shown in pink here. And we are looking at the olivary inputs coming into the Purkinje neurons, and that is shown in green here, both in early stage larvae, the 4 to 5 day old larvae, as well as the late stage larvae. And immediately you can appreciate that that there are inputs of variable amplitudes, as I mentioned before, and that the inputs are not strictly tuned to the timing of the motor response. And that is precisely what we observed. So here, let me show all of that at once. Yeah. So in the top panel here, 0 is the start of the motor bout, both in the green plot as well as in the purple plot. And in the green plot, you can see that the frequency, each dot is a CF input. And on the y-axis are the bouts that we recorded from all of the larvae that we recorded from. And the tick marks are separating the fish that we recorded from. So each tick mark, corresponds to one fish. And you can see that for all of the bouts that we recorded, that the climbing fiber input frequency overall is much higher in the early stage larva compared to the late stage larva. This again is reconfirming what I reported to you earlier. The second thing that we observed is that there is an increase in CF input frequency around the start of motor bouts, both in the early stage larva as well as the late stage larva, as shown here. But the tuning is much more broader in the early stage larva compared to the late stage larva. So there is more specific upregulation of CF input frequency closer to the motor bout start here in the late stage larva compared to the early stage larva. So that means that when a motor bout occurs, and within so these motor bouts in the fish are about 200 to about 600 700 milliseconds long. Sometimes they can be a bit longer, but most of the times it's under a second. And so when the fish starts to swim, it fully expects that its swim will have an effect on the sensory input coming in. It expects to compensate for the forward motion of the grating. And when that does not occur, then there is an error signal conveyed by means of the CF input. And that error signal is much more tightly coupled during the motor bout in the older larvae compared to the late stage larva. Now, what is also interesting is that the tuning, both in the early stage larvae as well as in the late stage larvae, is much better for the large amplitude CF inputs compared to the small amplitude CF input. So we arranged the CF amplitudes according to their magnitude. So the 0.8 to 1 is the top 20 percentile of all CF inputs recorded in terms of their amplitudes, both in the early stage and in the late stage. And the rest are the weaker, what we call as weaker inputs. And you can see that the timing of these weak inputs is all over the place, whereas the stronger inputs are much more concentrated near the start of the motor bout, which is marked here in these colored bars. And that is also quantified over here. So on the plot on the left-hand side is the large CF amplitudes, and the plot on the right-hand side is the small CF amplitude. And you see that for both, so the green is the early stage larva, as always, the same color code, and the purple is the late stage larva. And there is an upregulation of CF inputs as shown by mean CF frequency per cell near the motor bout in both the early stage as well as the late stage larvae. But if you look at the small amplitudes, the early stage larvae, the late stage larvae simply do not care. There is no upregulation, significant upregulation of CF frequency, but these early stage larvae, which are still undergoing synaptic pruning, now have an upregulation of synaptic inputs, even for the small amplitude. So it's sort of like a corruption of the signal that's coming into these Purkinje neurons, where the large amplitudes may be the ones that will stay later on after synaptic pruning, and therefore these are the ones that are conveying important information. In these, poly-innervated Purkinje neurons, the signal is corrupted by the small CF amplitudes, which are the ones which are ultimately going to be pruned away, but are present at these stages and perhaps prevent the Purkinje neurons from conveying meaningful information about the timing of motor bouts and the sensory-motor consequences of the motor bout. Okay, so that's a very longish explanation of this experiment. So to summarize this part of the talk, synaptic pruning fine-tunes sensory information and motor error encoding, and CF inputs to PN are more directionally selective after pruning, and motor-related CF inputs are also tightly coupled to bouts after pruning, and preferentially the large amplitude ones are the ones that seem to convey information about motor error during swim bouts. What does
Vatsala,
that?
I
Yeah,
have a question from JR. JR, go
yeah.
ahead.
Hi, Vatsala. This is Jennifer Raymond.
Hi, Jennifer. Good to see you. Meet you again. Meet you.
Yes. I was wondering if these are a motor-gated sensory input or a pure motor. If you record in the dark, do you see motor-related climbing fiber responses?
Yes, if we record from the dark, we do see climbing fiber related motor inputs. But in this case, it's a very good question. We cannot distinguish between the pure motor versus the motor related sensory input, so to speak. But in the I mean, it's still an error because it's a paralyzed larva, and so there is no proprioceptive information coming in, and that by itself could trigger an error. And so we have no way of distinguishing that. But having said that, you can record in this experiment, we are giving it visual stimulation. But in the earlier experiment, for example, here, my earlier slide, like these ones, these are recorded in the dark spontaneously. And so these would correspond to, and these as well, these as well. These CF inputs that I've marked here in red, these were recorded in the dark, and there are fictive motor bouts that are happening, and there are CF inputs that co-occur with them.
I see. Yeah, I mean, it's really very similar to what we see in the olivo-cerebellar of the mammal, where in the kind of behavioral or part, there's visual climbing fiber responses, but they also are activated in the dark. That was first shown by Jerry Simpson's group, and then we replicated in those too. But the question of like exactly what those non-visual signals are, I don't think we're quite sure in the mouse.
Isn't it amazing how much similar the fish and the mouse are?
Yes. Thank you.
So, so I come to
question. One more
yeah.
question.
Yes.
Thank you for the talk so far. And I think the optomotor behavior in zebrafish is extremely interesting. And it's reminiscent of the Genelia work in 2019, where there was this neural substrate for futility in open loop, where there was some integration of errors. There's a similar grading design, and they found that to be astrocytes that integrated this error, and then the fish would stop swimming. Have you found a similar, and I guess it's the plot right before this, neural substrate for integration of these errors that sort of prevents these complex spikes in maturity?
You're talking about the Mu et al. Paper from Misha Ahrens's lab, beautiful work. And yes, so it is true that if you keep giving the open loop, that they will stop responding. So that's why we give it in pulses. So we let the fish rest for a brief while before we give them the open loop again. If we were to give open loop continuously, they would stop responding. Now, whether, we have not done the experiment to see whether, in open loop, if you give continuous open loop and they stop swimming, how the climbing fiber inputs are integrated. Is that your question? Did I understand it correct?
Yes, yes, and the differential between the early and late, where there appeared to be like a continuation of complex spikes as if, or sorry, climbing fiber input as if, this error were somehow felt differently early versus late, where there was less climbing fiber input for late. I was wondering if there's some difference between the two.
Yeah, so no, we have not done that experiment where we show the open loop gratings continuously and let the fish, show this futility response. That would be something interesting to try out. But we have done something else which will bring together the aspects of sensory and motor tuning and how that leads to more reliable internal models of the world. So your question comes at the right time. So even learned futility is an internal model of the world, where you learn that, okay, your actions are not going to have any effect, and therefore you integrate that information over a longer period of time, and you make a model of the world which allows you to respond or not respond appropriately. Does that answer your question? Can I push ahead?
Of course, thank you.
Okay, so it is known that CF inputs trigger large calcium signals in PNs, both somatically as well as in the proximal dendrite. Our own experiments, for example, showed that, so we did in Purkinje neurons expressing GCAMP6S, we did loose patch recording. So we have a record of the calcium signal shown in green here simultaneously with the electrical recording, and we did observe very large calcium transients that are correlated with the electrically recorded CF inputs. And we can also see that over here. So this is, Purkinje neurons expressing GCAMP6S. So Aldoca specifically drives GCAMP expression in Purkinje neurons. We can be sure that all of these neurons are Purkinje neurons, and you do see very strong signals. So then this allows us to look at Purkinje neurons as a population and ask, at these different stages, early, intermediate, and late, as the synaptic pruning is progressing, what is happening with population encoding of various things. So it came in timely that another brilliant graduate student in the lab, Sriram Narayanan, had recently published work on a pattern learning task for larval zebrafish and how Purkinje neurons participated in encoding this learning. So what Sriram did was to place larval zebrafish under a custom 2-photon microscope. And beneath this larva, we showed the same black and white gratings. And there was a small window to film the tail movements of the larva. And now this we did in closed loop because we wanted to image for a very long time. And we wanted to make sure that the larva is responding all throughout the experiment. So what Sriram did was to take the tail movements and use that to drive the visual stimulus. So when the larva is moving forward, the grating will be suitably adjusted and brought back according to the speed of the tail movement of the larva, and therefore the larva would keep responding. And the task that we used was something which looked like this. So there are pulses of forward moving grating. This is just a schematic. So the larva is not responding here, but in real life it would. So there are pulses of forward going gratings. And then suddenly we will surprise the larva with something that's a deviant stimulus or a probe stimulus, which is a stimulus that's going backward, but also is much weaker compared to the forward going stimulus. And the reason we did this was that a strong backward going pulse would trigger a struggle response in the larva, and we wanted to avoid that. And therefore we gave something that was comfortably different from the forward going stimulus, but also did not induce any motor response. So to play that again, so you have forward going gratings, a number of them, and I'll tell you, how we varied this number. And then after repeating this stimulus for a little while, then we show a probe stimulus like that, right? And before we went further with the experiment, what we did was to show a scrambled stimulus to get the directional tuning of Purkinje neurons. So I already told you that climbing fiber responses to Purkinje neurons could be directionally tuned. And so what we did was show a combination of different flow stimuli, weak backward going like that, strong forward going, strong backward going, weak forward going, and so on and so forth. And we recorded the calcium responses of Purkinje neurons. And using regressors, we then classified neurons as either forward tuned or backward tuned neurons, or some were motor tuned. So this is the total data that we got in young, intermediate, and old larvae. And the numbers here correspond to the number of backward tuned neurons. That is what I'm going to focus on in the next couple of slides. So the top panel in each of these things, the top is the forward tuned cells, the middle is the backward tuned cells, and the bottom is the motor tuned cells. And what you see is that you have all these three populations of neurons. And if you take the neurons which are tuned to the backward going pulses, and after acclimatization, so I call the repetitive forward going pulses as acclimatization, you keep giving it repetitively. So the response to the same weak backward going probe stimulus before and after this acclimatization looks very different. Where in baseline, the response looks like this. After the larva has seen a series of forward going pulses, the response to that same probe stimulus is enhanced. That's shown in the black here, right? So this work was published earlier this year. So, I would urge you to go and look at it. This is in the 6 to 7 day old larvae. These are the intermediate stage larvae. And what Shivangi was interested in understanding was to do the same experiment, but at different stages of synaptic pruning, and ask whether this response looks different. And the reasoning is as follows. Through various experiments, Sriram had shown that these are predictive error responses. This enhancement that you see after acclimatizing the larva to a series of forward-going pulses is a prediction error response. And correspondingly, in the forward-tuned cells, there is a prediction response. And both the prediction error as well as the prediction response are important for modulating latency to the forward-going pulses. That's a lot that I've packed in one sentence. I'll unpack it as we go through the data. So what Sriram observed was that if you give 2x, 4x, 8x, or 16x pulses, as you increase the number of repetitions, the amplitude of this enhancement goes up. Okay. So you can see that over here, whereas for the 2x, there's not much of an enhancement that you see before and after acclimatization. As you give 8x or 16x, the amplitude of the enhancement goes up significantly. So that means that the history determines the response of these backward tuned cells to the same visual stimulus. In a sense, the expectation that you will get a forward going pulse modulates the response of these neurons to the same visual stimulus that they had seen before. So this is evidence of predictions that are happening within the larval zebrafish brain. So what Shivangi asked was, does synaptic pruning affect the way in which the larva encodes these predictive error signals in the Purkinje neurons? And she finds that, of course, they do. So here again, following the same color code, the green is in the early stage larva and the purple is in the late stage larva. The degree of enhancement, which is actually clearly shown over here, the degree of enhancement shown by these dotted lines over here is much greater in the late stage larva compared to the early stage larva. And we propose that this is because, of the sharper tuning to directional information that the Purkinje neuron gets during the late stages after synaptic pruning, that it's able to encode directional information correctly, and then that allows these neurons to then encode prediction errors reliably. And what is the use of all of this for the behavior? Because I had promised you earlier in the talk that we are looking at circuit implications and behaviors. So I said there is also a predictive calcium signal. And what, let me see, yeah. And what Sriram had found in his paper was that in the forward-tuned Purkinje cells, the calcium signal that occurs right before the start of the flow response itself is predictive of swim latency. So stronger the DF over F even before the flow starts, lower the swim latency. And that is shown in these plots over here. So Sriram had looked at this stage larvae, which is the 6 to 7 day old intermediate stage larvae. Both in the intermediate stage as well as in the older stage, there is a significant negative correlation between swim latency and the predictive calcium signal that we see in the Purkinje forward-tuned Purkinje neurons. But in what Shivangi finds is that the early stage larvae, there is no such significant correlation. And in fact, these larvae, these Purkinje neurons do not seem to have this predictive calcium signal. Corresponding to that, what we also find is that as we give repetitions of the forward-going pulses, the latency to swim goes down in the intermediate and late stage larvae. So they respond quickly, more quickly compared to when the trial started. But in the early stage larva, such modulation of latency is not there. So this points to a mechanism in which predictive calcium signals in Purkinje neurons are correlated with swim latency and drive the modulation of swim latency. Again, this is because, we had done some lesion experiments in Sriram's paper, which showed the involvement of cerebellum in the modulation of swim latency. And that's why I'm saying that it's causal, and that such mechanisms are absent when supernumerary climbing fiber synapses are present. So I've taken more time than I was asked to, but I will summarize here. I've shown you that larval zebrafish CF inputs to Purkinje cells undergo synaptic pruning in the first 2 weeks of life, similar to that observed in mammals. While pruning is happening, larvae exhibit multiple motor behaviors and use their cerebellum. When PNs are multi-innervated by climbing fibers, their sensory-motor tuning is more broad. And when they are mono-innervated, such tuning is very sharp. And this allows PNs to participate in motor planning and mediate faster behavioral responses. So with that, I will end my talk. These are the funders. And that's my lab before pruning. Some of these have escaped the lab, graduated and gone off to greener pastures. We need a newer lab picture with fewer members. Some are summer trainees who are no longer there. So we'll have a post-pruning picture taken soon. Thank you very much.
That was wonderful, Vatsala. So if there are questions, please just go ahead and raise your hand. I want to start by asking you about the data that you showed regarding even at late stage of development, the Purkinje cells can have 2 climbing fiber inputs. Have you gotten a chance to dissociate the visual tuning of these 2 climbing fibers independently and ask how similar they are?
No, we have not done that. Okay, let me actually go back. The one thing that we did was to look at in the case of the visual experiment, we actually recorded extracellularly, and we can only look at the timing of the CF inputs. The amplitude information is not so reliable because it's loose patch recording. But in the case of the motor tuning, we did look at the voltage clamp recordings of the EPSC amplitudes. And in that case, we were able to dissociate the large amplitudes from the small amplitudes. But yeah, we have not looked at the same thing, corresponding thing with the sensory visual input, if that's what you're asking.
Yeah. Thank you so much. So Jennifer, go ahead.
Those predictive responses are so cool. I was wondering if you can see anything if you omit instead of presenting the small backward, if you have forward, 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 and then just omit. Do you see a response?
So in the backward tuned neurons, we don't. So we actually did that as part of that paper, where we did different probe responses. Forward, give a series of forward going pulses, and then a probe which is a weak forward response. And also we did no grating, stationary gratings, bunch of other things we did. But in those cases, we do not see a probe response when we do that.
And do you need the time? Do you need it to be pulses of forward motion, or can it just be forward continuous for some amount of time and then give the weak backwards?
Very good question. So we did do that experiment. It is in that paper. So we do not see an enhancement if it's a continuous forward-going flow. And the reason is that, if you're thinking about accumulating evidence in a probabilistic fashion, if you give pulses of forward flow, then you're increasing the probability that the next pulse will be a forward-going pulse. Whereas if you're giving continuous flow, there's no information about what the future is going to be. And therefore, you do not see a predictive response.
And do you play around with the time of the pulses, like how frequent they
so
are?
In fact, yeah, very good question again. In fact, we first thought it's timing. But later on, we figured out that it was not timing. So we did do an experiment. This is all in that I would, strongly recommend.
Read your paper, yep.
So we did do the scramble timing where we gave the pulses at jitter times. And if you do that, you still see the error response. So it doesn't seem like the fish is tracking time so closely as much as it is accumulating probabilistic evidence of what the next stimulus is going to be.
Got it. Thank you.
Wonderful. Well, Vatsala, thank you so much. Are there any other questions? Wonderful. Just one clarification. The last experiment that you showed, I wasn't sure whether the motion of the visual stimulus was first, then the movement or of the tail, or was it the movement of the tail that initiated the motion of the visual stimulus?
So it's always the grating that starts moving first, and then with some latency, then the fish generates a forward swim.
I see. Thank you so much. Wonderful.
Well, thank you very much. I really enjoyed giving this talk, and thanks to everybody who's attended. And if you have any comments, please do write to me. As I said, we are writing this up, and we would appreciate any and all feedback that we can get in getting this beautiful work out.
It is wonderful. Thank you wonderfully for this. And I hope the feedback that you already got was useful to you, and have a wonderful evening.
Thank you so much. You too. Everybody have a good day.